It is Thursday, July 30th, and this is the Biotech Voyager. Thank you for coming. This is the show that covers the early stage signals inside of biotech. We're covering what's next, mapping the early stage signals before they become big news. Thank you very much for joining us today. We've got an exciting show at 12 o'clock. So about an hour from now, we'll be joined by Michael Torres. He exited Crossbridge Bio to Lilly for $300 million, and now he's going after longevity gene therapy. So very much excited for that conversation in about an hour. But between then and now, we've got a lot of really exciting signals to cover. So thank you for joining us. If you have any questions at any point, feel free to drop them in the chat. But yeah, Jeff, let's get into it.
Sweet, let's do it.
Excellent.
So just so you're aware, I've been sick for like 2.5 weeks. This thing is just like not going away. So if I seem a little scatterbrained, or if I have to like mute the mic to blow my nose,
we'll let you do that, Jeff. Not a problem.
Okay, just forgive me. Yeah, so biotech, let's do it. So not much, honestly, has changed with the top like 10 companies since Tuesday. What might be even like better, Ben, to do during this little section here is go to the recent signals. Because we can kind of get a deeper look at like what's been happening recently. We had a bunch so yesterday we had like 3 or 4 companies close seed rounds, which is cool for us working with companies in the early stage therapeutics area.
Yeah.
I see like seed round close and I'm like, ooh. Yeah, and then immediately I'm like, cha-ching, cha-ching. They have money now.
Good for them.
So that's been good for us. So yeah, things like and then some grants coming in. Shape Therapeutics, Michael J. Fox, which I think Extosis did the Michael J. Fox grant closed recently.
That's right. Michael J. Fox has been on a rip. We'll talk about that a little bit later.
Yeah, and then I added this one, and this company is like barely known, Sinolo Therapeutics. They appointed their CEO yesterday, which I'm super excited to see because I've been watching Anish Suri is the CSO of this company. He came from Q Biopharma.
Okay.
Which Q is like a really cool company. But they appointed their CEO yesterday. So they're probably going to be preparing to come out of stealth, I would imagine. They're from RA Ventures.
Okay.
Good so, which I think is like a venture studio that's associated with RA Capital. But they're big time.
That'd be hilarious if they're not. They're just like, we're going to rip off their name, but it's going to be slightly different.
Yeah, I think, yeah, it might not be. I don't know. But in fact, I'm pretty sure that the profile says like RA Ventures, RA Capital Management. Yeah. Okay. So
one and the same.
Yeah, but I think that during the section for Sinolo, I want to like look at Q Biopharma because they're not like super similar companies, but Q has like the coolest T-cell engager technology that I've ever seen. And I'm not just saying that.
Excellent.
Probably kind of cooler than Deck Bios. I hate to say it, but Deck Bios there's different approaches. Well, you'll see what I mean. So yeah, here's your boy, Insilico Medicine, fast track. They're just like, everything's getting fast track. Everything's getting orphan drug. It's like, they're insane.
We will visit this story in further detail later.
Okay, perfect.
I want to get into the 3 different components of AI that they are just absolutely dominating in and how it allows them to do this.
Yeah, that's actually going to be, okay, cool. Looking forward to that. So yeah, if you want to like jump into it, we can just jump in.
Perfect. All right. So yeah, the first one of the day is going to be Aractogen, Jeff. So I believe this one is yours.
Mm-hmm.
So let's do it.
Yeah. So we're going to learn about this company together. Because the reason that I picked this was one, they closed, I think, $31 million. And it seems like it's a Chinese company.
31 million.
Yep.
Right on.
31 million. And the difference here, when you think of like an RNA-based therapeutic that's going to influence the expression of a gene, not a gene therapy, an RNA therapeutic that influences the expression of a gene, what do you typically think of?
Transcription factor, likely.
I think of like an siRNA or an ASO, like degrading RNA.
Sure.
Right. Well, this is small activating RNAs, which actually are like, it's like an siRNA or an oligonucleotide, right? but they're meant to increase the expression or increase maybe the stability of certain RNAs.
Interesting.
So it's like the opposite of what you would think. And if you look here, it's for cancer. And what they do is they increase the expression of tumor suppressor genes to suppress tumor growth, P21 being one of them. So,
Right.
or I guess,
Okay.
yeah.
So this is interesting then. So if we take a moment, right, when we think of something, when we think of these different tumor suppressor genes, right, probably one of the just very large mechanisms that allows cancer to progress in the 1 in a million chance, right? You know, you and I have gotten cancer in this conversation already, and it's already been dealt with. Hopefully.
I'm not sure it's that frequent, but
I've heard it's like once every 15 minutes.
what? Okay, we
Yeah.
can return to that. I don't know where
Yeah.
you heard that.
But anyway, yeah, no, this is super interesting, right? Like so many. Basically, I'm just trying to what I'm trying to emphasize here is so many different things have to go wrong in order for you to get cancer. And one
Yes.
of and just and one of those key things is going to be you need to coordinate the downregulation of all these different suppressor genes that are designed to help detect and eliminate cancer. And so this being one of them, it's super cool to see an approach where instead of going after uh one of the actual oncogenic genes, it's very much the opposite. It's like let's enable some of the indigenous fighters, as it were, to become even more powerful so that they can start defeating this from within.
Yeah. And like this seems like a one of those things where I read it and I'm like, duh, why hasn't anybody done this before? Like, just why? Why don't we give, like, for example, overexpressing, let's say, RB1, which is a massively popular like tumor suppressor gene, wouldn't do much. Like if a tumor suppressor gene is like completely lost or mutated in like a cancer, overexpressing it just like systemically probably wouldn't. I feel like I should know this being the cancer guy, but it's just like, why can't we just overexpress P21 just like systemically? Because it probably has to do with like wound healing and like we need proliferation of our cells normally, so you can't just overexpress P21. But it seems to be one of those things where like, yeah, just if we can get an intratumoral, like a localized expression of these things, why haven't we done this before?
I'll bet.
Um,
I'll bet you. All
so
right. So here's my thoughts on probably what's happening. This is going to allow it to be very specific to where cancer is taking place, because I would imagine, right, there's some sort of detection event that's going on. It's like, oh shoot, cancer is here. Ding 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 ding. Let's get this P21 up and running. And in the context of the cancer microenvironment, that gene is getting actively downregulated in a situation where it wouldn't be in normal cells. In normal cells, P21 isn't getting activated at all because nothing is going wrong. P21, the alarm bell is ringing, but no fireman is ever showing up. And so this might be a way to very specifically only allow P21 to get further activated in the context of cancer cells without any off-target effects in normal healthy cells.
So yes, like P21 is a downstream transcription target of P53. And P53 is like the tumor suppressor gene, right? So yes, it would be the kind of thing where it's like, hey, we just need the right now we have this is like in a cage, right? And if you take the cage, it's kind of the difference between like, hey, we have this thing in a cage, but we need the thing in the room, right? If you just put more things in the room, taking the cage away lets the thing go into the room when the room needs it, right? Versus just stuffing it full of it regardless. So yes, 100%. Now I'm very interested to know how these things work in particular. So they call them small activating RNAs, saRNA, right? If you go to their website, let's go to their science page real quick. RNA activation. RNA-A uses short duplex RNA known as saRNAs to target and turn up transcription of an endogenous gene, leading to restoration of endogenous protein function. See, okay, hang on. Okay, saRNA is loaded into the AGO2 protein and processed into a single strand. That's fine. Enters the nucleus and recognizes a non-coding regulatory sequence embedded in chromatin proximal to targeted gene. Okay. Okay, this is significantly more complicated than I thought it was going to be. It looks like it's not, it's pre-transcriptional regulation. It's not
I'm
post.
seeing this, yeah. So it's not quite what I was thinking it was.
Yeah, which is even that's really complex. Like that would mean that the actual the pathology, like the pathological or the reason that P21 is not being expressed is because of some aberrant, like, what? Okay, this is hard to figure out in real time. But apparently I thought that it may have been a thing like that, where it's like we're going to post transcriptionally help the RNA become more stable or whatever. But the thing is that if P21, because of like a P53 mutation, if P21 is not even being expressed, how does this help that? Right? Like if P53 is needed to transcribe P21, no amount of like help it become transcribed is going to help because P53 is not there. So there's obviously some kind of nuance that I'm not.
Okay. Wait. Go back for a second, because it says, and it recruits additional proteins to form the RITA complex. Okay. So maybe that's part of the issue is in cancer cells, we don't have the same capability to bring together the RITA complex in order to get this thing up and running.
You know what this might look like, actually?
What does it look
this like? Hang on a second. Okay, this is actually super sick.
Illuminate us, Jeff.
This is basically a programmable transcription factor.
Ooh, yes, please. Okay,
it's
so
like
this is kind of what I was talking about earlier.
It's like a transcription fact. You can basically say, hey, here's this RNA molecule that will go and bind to wherever you've kind of made it complementary to, and it's going to promote the formation of a RITA complex, apparently, which might, I don't know what a RITA complex is, to be totally honest, but it might be involved in like the nucleation of the transcription complex, which is what transcription factors do, right? Like, so if you can target certain regions of the genome with RNA-A or saRNAs and promote the recruitment of transcription complexes, you've effectively created a RNA-guided transcription factor.
Super cool.
Which I guess is like kind of the opposite of what like Scribe Therapeutics is doing, kind of, in effect. Like Scribe is saying, hey, here's this guide RNA, go and bind here and recruit like transcription repression complexes. This seems to be kind of the opposite. Go and bind here and promote transcription from that locus.
Interesting.
Really cool. If that's what it is. Yeah.
Well, folks, you may have heard about saRNA for the first time here on the Biotech Voyager, because we are.
Yeah, it says formation of the RITA complex augments steps of transcription initiation and elongation via RNA pull 2, enrichment, phosphorylation, and epigenetic modification indicative of a relaxed chromatin. So it literally is like a programmable transcription factor.
Phenomenal.
Really cool. Yeah, see that?
All right, so
interesting.
I should do a standalone video, or we should make a graphic about this, Jeff. The difference between saRNA and siRNA, because that would be so handy.
I wouldn't even be sure if the difference between saRNA and siRNA would be the right video. It would more be like this company is making RNA guided transcription factors.
RNA guided transcription factors. That's a new one. Or at least
really
it was.
Really interesting. Yeah, I haven't heard of RNA. I'm in like cancer genetics, I guess you could call me that. I've never heard of RNA-A. So maybe it's a new thing that they've kind of developed. Anyways, didn't expect that to happen.
Anyway, they've got $31 million, so clearly there's investor interest in this kind of new approach. So it is very good to see.
Yeah, and investor interest makes sense because this would be the kind of thing that could be used across all kinds of different indications. like
Right.
across different disease sectors. Like they've got one for ALS and then one for cancer, bladder cancer. Yeah, that's cool.
Nifty. Okay. And whereabouts are they based?
Xingzhou or Suzhou. I can never pronounce these things the right way. They're in China.
Yeah. I think it's Shuzhou.
Shuzhou, Jiangsu.
Yeah. Well, kudos to them. Glad they've been able to get a bunch of money. And yeah, they're driving 4 different things through the clinic by the looks of it.
Yeah, this is non-muscle invasive bladder cancer and then ALS, it looks like. And then they've got a couple of collaborations. What is this one for? Duchenne? Which, what?
Wow.
Okay.
Ractogen. Well done.
Yeah, ALS. Cool. All right. Anyways.
Phenomenal job. Okay. Well, that's gonna be a hard one to beat. uh, anyway, yeah. So, folks, we're gonna be moving on from this one on to the next story of the day. But we're gonna be having Michael Torres, the CSO of Artan Bio, at 12 EST. So feel free to stick around for that. And if you have any questions either during or before his interview, feel free to drop them in the chat, and we will get to them post haste. All right. So next up here, Shape Therapeutics. This one's mine, Jeff. We can run over to Shape Therapeutics.
So
okay. So
yeah,
a couple different things here. Michael J. Fox Foundation has been on a little bit of a rip recently. This is going to be a little bit controversial, but one of the big challenges that I think the Michael J. Fox Foundation has run up against is the fact that despite all of the large amounts of money that they've poured into Parkinson's disease research over the last, 25 years, I think, however long the Michael J. Fox Foundation has been around, they haven't really been able to get super far given how long they've kind of been in the space and how much money they've poured into it. And so what I see them doing right now, which we want to very much applaud, is they're like, hey, we need to start funding some radically different approaches here. What we've been trying hasn't been working super well. And so as the Michael J. Fox Foundation, we need to do a better job of finding these really novel approaches and funding them and getting these things forward. And so a couple weeks ago, we had Extosis on. They had also received a very large Michael J. Fox Foundation grant. They were focused on a small molecule for mitochondrial health, if I remember correctly, Jeff. And so this is kind of on the opposite end of that. And maybe it's kind of like a combination therapy play as well. So Shape Therapeutics, on the other hand, is a gene therapy for Parkinson's disease, which is really nifty to see. Again, like, kudos to the Michael J. Fox team that you're funding all sorts of very different approaches to fix obviously a very devastating disease. So yeah, let's just kind of dive into some of the key details here. Again, what we're focusing on here is a disease, a genetic, let's do this again. A gene called ASYN. ASYN is the target that they're going after. So it's an upstream target. So instead of blocking the protein after it's made, they go earlier, reducing how much of it gets produced in the first place. And they're using RNA editing. And so they've designed and optimized the editing payload and the delivery vehicle. And so this is an AAV delivered, AAV single dose delivered therapy that does RNA editing for this ASYN gene that's affiliated with Parkinson's progression. I'm trying to figure out what this molecule does. Okay. So yeah, the key culprit in Parkinson's disease is a protein called alpha synuclein, or ASYN, and it misfolds and clumps in the brain, which ends up killing neurons. And so the goal would be, okay, can we basically go back, use this RNA editing technology to prevent ASYN from getting made as much? And basically what they're focusing on here is an AAV delivery, which is super cool. And what on top of that, they are developing 2 different approaches in terms of how they administer it. They administer it inside of the brain, is my understanding, and then they're also they're also delivering it systemically as well. So it's a really interesting combination here.
Yeah, I'm trying to figure out. First of all, their website is insane.
That's
Like,
really
what?
beautiful.
Um,
we
but
see
I'm
we see
trying to
we see a
figure
lot of
out.
bad biotech websites. This
Yeah,
one's beautiful.
I'm
Well
trying to
done.
figure out like what this does. Yeah, they're saying that it will. They have an RNA fix
Yeah.
shape therapeutics AI engine to analyze massive data sets. Combines AI and RNA technology. I cannot figure out what it does. Leveraging AI and RNA technology to discover programmable RNA medicines. Okay.
Okay.
We
So
use
yeah, let's go
RNA to
here.
design novel RNA. Okay. The language is really, really general. Recruits ADAR, a naturally occurring enzyme found in all human cells to correct the body's instructions for making proteins by recoding RNA without permanent changes to the DNA. RNA fix can be applied to correct mutations or purposely create them to treat a vast array of genetic disorders. Oh, that last part
Come on.
there.
Let's go.
Purposefully create them, as in like introduce a stop codon or something to create.
Yeah.
I wonder if that's what they do with synuclein.
Must be. Right? So again, alpha synuclein is something that you don't want. It's a misfolded protein that's clumping and killing neurons. So if you can basically go back in without, doing some sort of permanent gene edit, but basically just as soon, it's kind of like almost like spawn killing. It's as soon as they come out, they just stop it from getting made, or they're editing it to make it the correct version.
Yeah. So the like spawn killing would be more akin to like siRNA, right? So you're
Right.
just kind of like, as the RNA is being made, you bind to it, cut it up, and it's gone. This is kind of like it can do more. It's more like versatile. Like it will recruit the ADAR enzyme, and like there's all kinds of companies that use the same kind of technology. It's RNA editing
Right.
technology, right? Um uh and they're doing it to like correct mutations or to introduce them, which, yeah,
Interesting.
I mean, you can think of all kinds of like genetic mutation driven pathology that could be applied for. Parkinson's disease apparently is one of them. Although I guess like it could be used as like an mRNA, in effect, an mRNA degradation mechanism as well. If you introduce some kind of stop codon or something, um, like you can just intentionally create truncated proteins, which in effect downregulates the expression of the protein, um, which I'm going to guess is what they're doing here. But the
Right.
technology can also be used to correct mutations or to introduce ones that aren't even like stop codons. You can introduce a mutation to create some kind of like different thing. I don't know. But it allows you, that's really interesting. It allows you to kind of have a lot of like versatility there. That's cool. I'd be
Well
curious to
played,
know.
Shane.
I doubt that their website goes into as much detail as I want here, but I'd be curious to know like what are they doing to the alpha synuclein RNA that makes it treat the disease.
Right.
Um, so what did they do? They got a grant from the Michael J. Fox. Do you know how much it was for? Did it say?
I believe it's for a couple $1,000,000 at least.
Yeah, typically they're like around like 1 to 3 mil.
Right.
But cool. That's a cool
Man,
company too, man. Dang.
just so many. This is wild. We're really are starting to see an emergence of a lot of different like RNA editing
Yeah, yesterday was
technologies.
or Tuesday was a lot of like T cell engager stuff. Or no, was it Thursday? Last Thursday was a lot of like no, it was Tuesday. It was a lot of like AI based analysis of transcriptomics to create therapeutics.
Yeah.
Today it seems to be a lot of like RNA based therapeutics.
Well, yeah. Super cool to see. Well played to Shape Therapeutics, and again, kudos to Michael J. Fox Foundation for funding some bold, novel approaches. We wanna commend them for that.
Man, this cold
Excellent.
is kicking my butt, man.
Oh,
It's been going on for too long.
you're doing well, Jeff. All right, but we are on to Sinalo Therapeutics, I believe, Jeff. This one's yours.
Oh,
Uh,
are we already there?
we are.
Oh wow. Okay. So there's not a whole lot of information on this company because look at their website. Their website is like really minimal. Advancing a new class of therapeutics for cancer launched by RE Ventures and leveraging a proprietary discovery platform, we are rapidly building a pipeline of differentiated, conditionally active immunotherapies which promise to be highly effective and safer than the current standard of care. Okay. The thing that catches my eye here is oh my gosh, where did it go? Why am my brain is just like not working? Conditionally active, logic gated immunotherapies. Um, Again, I don't know what they're doing, but I do know their CSO, his name is Anish Suri. You can find him on LinkedIn. He has no profile picture, but he exists. He's from a company, or he was at Q Biopharma. He was the CSO of Q Biopharma for a long time. And I love this company. I love Q Biopharma, which is why I'm very excited to see what Sonolo is doing, even though I have no clue what they're doing. But Q Biopharma, do you know about Q? They've been around for kind of a long time, like maybe since like
I've
2015.
certainly come across the name, but I wouldn't say I'm very well versed in what they're doing.
It's insane. I think it's super cool. Now, therapeutically, I'm not like the expert in the space, right? But what Q Biopharma, look at these things, dude. It's insane. And again, this is not Sonolo, this is Q, but it's the same. The CSO from Q is now the CSO of Sonolo. So they changed their website up a lot. Huh?
So that should give us a clue of what's going on.
Yeah, it's going to be cool immunology. They don't have any pictures of their stuff. The Q Biopharma, come on, man. They had a cool website. They must have changed it recently. I wonder if I can find like, let's see. This is a little dangerous. Going to images here. Nothing. That's insane. I'm going to make him one. Oh, here we go. Hang on. Here we go. Okay. This kind of gives you an idea. So what they have, this is Q. What they have, this
so
is
just
like
to clarify, right? We're talking about Q Biopharma because key executive from Q Biopharma has gone to Sonolo Therapeutics, which could be a really key indication of what this really early stage company is going to be working on.
Yeah, really cool stuff happening at Q. So like the CSO is now working at Sonolo. I can only imagine what he's got cooking up over there. So this is what Q is making. I am very familiar with their 500 and 100 series. I don't know what the 400 series is. I don't know what it is. So, but their 100 series, it's two things. It's a peptide MHC loaded viral antigen. Okay, that's what this blue part is. And then these orange little gems are IL-2 molecules. Okay, so it's basically an IL-2 therapy. And if you know anything about IL-2 therapeutics for like cancer, you know that they're super toxic. They don't work very well because they're toxic, right? You give IL-2 or like interferon alpha, and you just get like CRS, right? So you can't just give systemic IL-2. But what you can do is give IL-2 that's going to go to a specific T cell population, right? And get a robust but not systemic activation of T cells. So what they do here is they will load in like a tumor antigen or like a viral antigen that they know is associated with the tumor, and they will give this to the patient, and then this right here will promote this IL-2 molecule basically to bind to only the tumor reactive T cells.
Okay.
Right. So you'll have tumor reactive T cells that are kind of there. They have their TCR that recognizes the tumor, but they're being suppressed, PD-1, blah, 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 blah. You put this in, this will bind to their TCR and then activate IL-2 receptors and just juice up that T cell. Right.
Nice.
And their Q500 series, this, oh my goodness. Yeah, this is a CD19 T cell engager for like autoimmune disorders.
Nifty.
Okay.
Autoimmune. Interesting.
Yep. And this is kind of the same principle. Okay. So these right here are short chain variable regions, fragments that target CD19, which is like, imagine that this is like what a CAR T cell targeting spot thingy for CD19. it's the exact same thing. So this is going to bind to CD19, the purple part, and then this part again binds to like virally activated T cells, which you have all over your body. So they'll use a viral antigen that they know a lot of people have like reactivity to, like, I don't know, coronavirus, whatever, right? Everyone's got memory T cells to some viruses in your body at all times. Well, you can just bing, 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 trick your immune system into thinking, oh my gosh, we have that virus around. Let's kill whatever we're bound to, CD19.
Nice.
And it kills B cells. So this is what Q is doing. Imagine now what Sonolo is doing. Like the CSO goes to Sonolo. They got conditionally active immunotherapies. And it's like, this is his new pet project. Like, I'm super pumped to see what they're doing. And I have no idea what they're doing because they're so early. They're so early. But also, they came out of RA Venture. So
right
like
there. That's some great
validation. Yeah.
He's got something underneath the hood that that made Peter Kolchinsky go,
Yes. So what the heck? The Biotech Voyager obviously has no clue what they're doing either. It's just like, here it is, whatever
just
this
put
is.
some crazy lightning things on it. Make it go wild.
So, including T-cell engagers, engineered for disease-specific selectivity across oncology and autoimmune diseases, aiming to improve efficacy and reduce off-target toxicities. Yeah, so keep an eye on Sonolo, even though there's like nothing to keep your eye on right now. But you can find Fawad Naumani on LinkedIn. He's there. So yeah.
Does he have a profile picture as opposed to the CSO?
He does. Yes. The CSO, Anish, Anish is actually like very active on LinkedIn, just with no profile picture.
That is a choice.
And I've told him, like, dude, come on, just do it. So whatever. But yeah, really. Oh yeah. Yeah.
Well, Jeff,
several times.
Get the CSO on the show. Let's see what's going on.
I'm not sure they're going to want to disclose information yet.
Oh, well, they could speak around the problems. Like, all right, this is a little bit of a rant. This is a problem that we have inside of science. You can speak around the problem, and you can kind of, you can talk about something by showcasing the shadow, and you're not revealing any of the details of the thing that is casting the shadow, but people can get a good idea of what you're working on and not be able to steal it or anything. And no one's really. If
there is any consistent quality among especially lab scientists, it is paranoia. Paranoia of getting scooped.
And I don't really think it exists. I don't think there's anyone who's like, do you know what? I'm sitting down doing my own lab research. I read a social media post, and I'm going to just stop all of my existing work and go try to scoop the other guy.
Well, here's chew on this. I have spent the past year and a half or 2 years learning how to build AI apps, for example.
Sure.
Right? I've gone through all the hard work. I fell over, the sticks in the road. I've had to build bridges, I could catch you up in like a week, right, giving you the roadmap. That's kind of how it is here. I spend 6 years in lab experimenting, wasting money, having negative results. And then here's what I finally figured out. Bing, there it is. You could reproduce that in a couple of months if I gave you the roadmap, right? So it's kind of like that. It isn't that no one's going to come in and take your idea. It's more like if they wanted to, they could reproduce what I've done in the past 8 years in like maybe 3 weeks if I tell them what to do. So there's that.
I suppose that's fair. And one of the concerns I have heard from folks as well, right, is actually China does this to some extent.
Yeah, IP right? Theft is like their middle name.
Well, I think I forget who was talking about it, but I saw a really interesting clip with regards to this where it's like we've got to figure out a new way to do patents, because basically you just have these massive data scrapers that are just scraping all new patents every single morning and trying to like reverse engineer it. We need to have a way by which you can keep your IP safe without publishing it to the world. So
I don't know the first thing about doing any kind of IP protection, but I agree. Did you see? I'm not super sure how like true this is. It could just be like fake, but somebody was using a Kimi K3 model in their app, and they were talking to it, and the person in the app called their AI Kimi, Like, hey, Kimmy, do this. And it was like, not sure who Kimmy is. My name's Claude. It's like, oh,
Oh,
is it now?
I see.
Yeah.
Interesting.
Yeah.
Uh-huh.
So
Yep.
again, could have been fake, could have been a joke, but also like probably true.
Yeah.
Mhm.
Yeah, this is unrelated, but the other thing I saw recently as well, I was watching TVPN, and they were talking with a guy with regards to like over the next 6 months, we should hit RSI, which is recursive self-improvement of AI models, and how that's just kind of like that could be a match that kind of lights this off too, in terms of where I will be able to go. So yeah, lots of different moves inside of the AI space that we are not necessarily covering here on the Biotech Voyager, except for this next story, which
Wow, what
very much
what
is
a segue.
what a segue. Insilico Medicines, a fan favorite around here.
Now, I will say that Insilico Medicines is probably on the border of being like emerging biotech. It's actually a little bit silly that I've got them in here, but whatever.
I've made a special request because I would argue they are still early stage enough just because they are on that cutting edge frontier and it's not like big pharma has bought them. They are their own thing doing some absolutely phenomenal work. So yeah, what I wanted to spend a little bit of time on today, Jeff, would actually be going through well, first of all, I guess the signal that we want to talk about here, right, is that Insilico Medicine itself has scored an FDA fast track for one of its molecules. I'm forgetting the indication for
Mesothelioma.
mesothelioma.
Mesothelioma. Have you not seen those class action lawsuit commercials for asbestos, Ben?
No, I'm Canadian.
Oh,
We're not allowed to
man.
have those on TV.
There's been a whole bunch of class action lawsuits for people who get mesothelioma of the lung or the stomach from asbestos in their homes and stuff.
Oh, so this is
it's
why the FDA is like, this is really important. We need to get a drug like this approved
maybe.
post haste. Interesting. Okay, so yeah, this is what this is what the Insilico Medicines is focusing on. But Jeff, if you can go to the press release itself, and actually click on that signal, I think it has in there the 3 different sections that I want to focus on. So what I would like to spend a little bit of time focusing on for Insilico Medicines are basically the 3 different pillars that make them a phenomenal company. So if you're not familiar with Insilico Medicines, please go check them out. They are phenomenal. Have basically they're one of the best in the world in terms of being able to identify and make AI designed molecules. And I want to take a little bit of time to go through the 3 different pillars that allow them to do that really, really well. So I believe it's found in this press release. So if you scroll down just a little bit, we're going to focus on their 3 different platforms that they're working on. If you scroll down past this, I think it's about Insilico Medicine. Is it in here? Maybe not. Back up for a second. Oh, do you want? It might actually be in the Biotech Voyager signal, and I went to the wrong spot. So if you go back and if you just scroll down here, back up, scroll back up a little bit.
They're right here.
Perfect. Yes, this is what I want to focus on. Okay, so there's 3 different things that Insilico Medicine is basically running literal thousands of different AI agents on, and it basically comes down to 3 different buckets. There's probably more, but these are the 3 that you really want to be focusing on. The first one is Pandaomics. Okay, this is where they're doing an incredible amount of work in the target discovery space. They are going through all of the research. They're running autonomous experiments. They're trying to figure out what is the best possible target for the disease that we're looking at. And so they can go run all sorts of different agents through all of the existing literature on this topic. They are then able to also start doing some different studies inside of the lab and like modeling all sorts of different scenarios and whatnot to figure out what is the best target for this specific disease. So that's the first one. The second one here is their Chemistry 42. So this is an entirely separate thing that is now feeding off of the first pillar, which is now molecule design. Now that we've identified our top target, or our top 3, or 5, or 6, or 15 different targets, now let's go and figure out what is the optimal molecule that we need to design to influence this target in the way that we need to. And they're doing, again, all sorts of crazy research in the background and all sorts of different AI design and like virtual simulations and whatnot to figure out what is the best possible molecule that we can design from scratch. Maybe it's never existed before in nature, that's going to very specifically influence the target that we've identified to move forward. And then, and this is what I think is so cool as well, their third one is Medicine 42, which is the clinical insight. So basically, they now figure out, okay, we figured out our target, we figured out our molecules. Maybe we've got like 100 different candidates across those different things for a specific disease. Now we're going to run this through the filter of the actual clinical trial design. Let's now kind of simulate which of these is going to perform best in clinical trials across all of the existing data that we already have about how clinical trials work in this area, identify any risks, figure out what we should be focusing on, and see if we can also basically prioritize these list of candidates that we've been able to develop so far to figure out which one has the best chance of doing well in the clinic. And as a result of that, they've spun out, I think they've got like 36.
It's something insane.
Yeah, it's like 30 to 40 different clinical stage assets across all of their different.
They might have like 3 in the clinic.
I think it's more than that. Sorry, like, yeah, I want to be precise with my. They have made a boatload of different therapeutic molecules. I don't know how many of them are in the clinic, but like really solid ones that are ready to go. And so they do a lot of out licensing to big pharma, to different groups, and that's kind of what we've seen as a string of different signals for them recently.
So the reason that I had the number 3 on my mind is because they recently advanced into phase 3 with their lead candidate, the TNIC inhibitor. But they have got about 10 in clinical trials and over 40 total programs. If you go to their website, like I'm going to go to this other screen here. I'm going to make sure it's still the same thing. Their website looked like the Matrix for a long time, and it had just like it was like going to like a SaaS platform. Yeah. Okay. Hang on. It's still that. Hang on 1 second. This is ridiculous. If you haven't gone to their website yet, like it's actually like, what am I looking at? Okay. Look at this. So you actually like can't really get a good idea because it's like super. Oh, here we go. Can you see this?
Yeah.
Okay. Like this is what their like biotech website is. This is their pipeline here. Right. And it's all like interactable.
It's
right.
Massive. Yeah.
It's huge. They have a huge pipeline. But you'd think, this is the thing. It's Insilico, right? So you can kind of just like have these things being discovered and tested in your sleep, right? And then
exactly.
we have 35 candidates. Yeah.
Yeah.
Yeah, I think, not to like downplay kind of what you've just gone through, but I think that a lot of companies have this same kind of workflow. Like an AI-based drug discovery company.
They're the pioneers here, though. They've just been able to generate so much more data. They've got then like autonomous labs hooked up to all of this, Jeff.
That's insane.
Well,
yeah.
Like I don't know if you can quickly like find a video of them kind of like walking through their autonomous labs that are based over in China.
So
they look like something out of Star Wars.
It's probably in their media section, maybe.
It might be.
But I've seen like. Maybe this is it.
It might be.
Let's try and find a spot. Can you hear it or no?
I can't hear it. No.
Okay. We've seen that video though before where like it shows like it's like a big, huge like robotics lab, all that's like, designed or pre my brain, dude. This cold is just frying my brain. You know what I'm trying to say? Ay.
Regardless, yeah.
I was gonna say predestined, like I'm in the Bible.
they've got some absolutely crazy labs that are just they've got thousands of different research agents and roles inside of their organization that are hooked up to a fully autonomous lab doing all sorts of crazy stuff. And yeah, they have one of their AI designed molecules in phase 3 for IPF.
Yeah, crazy.
It's crazy. Nuts. And the other thing that I think is also really interesting that maybe doesn't get as much attention about their whole approach as well is they're all focused on small molecules. And so I think that's going to be a really interesting play over the coming years for combination therapies too.
Well, it's also like their innovation is like on the computer, quote unquote. And then manufacturing a small molecule is like dead simple. usually, right? Versus like a cell therapy. I mean,
Yeah.
so yeah. I mean, they're obviously massive, like a gigantic early stage company. They're phase 3, Ben.
Exactly. Yes. Well, folks, in just about half an hour here, under half an hour, in 15 minutes, good gracious, time has flown. We are going to be talking with Michael Torres. He is the CSO of Artan Bio, focused on a longevity gene therapy. And if you recognize Michael Torres' name, he previously was the CEO of Crossbridge Bio, and it got acquired by Lilly for $300 million. So it's really cool to kind of see him switching from bispecifics to longevity gene therapies. Love to see that that progression. So before he comes on, though, we do have some additional signals we want to get through.
So real quick, though, Ben, does Artan Bio position themselves as a gene therapy company?
Yes, that's my understanding.
Really?
Yes, sir.
Okay. I'm just wondering because like it's a tRNA therapeutic. We're gonna we'll hear more about how this thing works later, but like it's always funny to see, like for example, you could technically call like a CAR T cell a gene therapy.
Some people do, and
right? Yeah, it's like come on, it's a cell therapy. So this is like more of like an RNA therapy, or like what would it even be? It's a tRNA, technically speaking. So for me, it's like it's an RNA therapeutic, not a gene therapy. But okay, anyways, who's up next? Is it Aspen?
It is Aspen. So it's really cool to see some of the announcements coming out from the FDA, because Insilico just got their FDA fast track, and now we've got an RMAT designation for Aspen.
Indeed. So Ben, go.
Dopaminergic.
That was good. That was good. The first try.
I've struggled with that word for many moons.
For those of you who don't know, Ben literally spent, I think, like a whole minute trying to say dopaminergic one time. That was, and I think it may have been for Aspen. It may have been for this signal right here.
It might have been.
Yeah, that's funny. So what they're doing is they're using iPSCs to create dopaminergic neurons for Parkinson's, to release dopamine. And it's a neurotransmitter, right, for Parkinson's disease. And I think it was an autologous iPSC. Yeah, which is crazy. When I think of iPSCs, I think of allogeneic, 100%, right? But an autologous iPSC, which means so where do they get the cells from? The skin? Reprogramming a patient's own skin cells, yeah, into dopaminergic neuronal precursors to replace the neurons lost in Parkinson's disease. So RMAT designation. Oh wait. Completes dosing. Was it this or we got a refresh. Sometimes for whatever reason, you see that?
There we go. Hey.
Yeah. Okay. So RMAT designation for saziniprocel. Yeah. So cell therapy iPSCs for Parkinson's disease. I love to see cell therapies for neurology. It's super cool because we're making neurons finally, kind of, sort of. I think that these things are basically just meant they turn into these dopaminergic neurons and just kind of like sit there in their natural home and just like release neurons. So it's kind of like a cell therapy, like drug factory drug, right. Sort of. They're not like transmit. You're not going to like be able to put these things in and get smarter. Right. In that sense.
Yeah.
But I'm waiting for the day, man, where you can like literally like regrowing neurons to make us smarter. Like I drank too much in grad school. Give me a pump full of neurons and help me recover my lost brain cells. Not quite yet.
I don't think that's ever gonna happen.
Yeah, no, I'm a lost cause for sure. But anyway, so stem cell biologist Jeanne Loring and Andre Bratliel, so Ben, explain to me, like I'm 5, the different FDA designations. I understand orphan drug. I understand fast track. What is RMAT designation?
So this one is I'm gonna mess it up, but it's more focused on like a manufacturing process.
Oh.
And so what it allows you to do is basically have like a much faster interaction with the FDA. Once you've got this manufacturing process in place, when you make additional therapeutics through it, you don't have to start from scratch. You're already basically like pre-approved with the FDA. And so you can just keep going again and again. So some different organizations like Solaris, for example, right, has RMAT designation now. Seleno also has RMAT designation, right? Basically, you don't have to start from scratch with the FDA, and they're like, "What is this whole manufacturing program that you're making, huh?"
Got
They've
it.
already done all of the pre-work, so it makes it a lot easier for early stage therapeutic companies to get through manufacturing a lot faster.
So okay,
It's
then
also applying to like some analytical instruments as well.
how would a company like
Nanomosaic has one
Nanomosaic has one too?
for their Tessie system. Yeah,
So then explain. Okay, this is. Oh man. Okay, they must be doing their own in-house manufacturing then. Aspen.
they must be.
Hmm.
Yeah.
Weird. Okay. Yeah. I
Well,
mean,
if it is
regardless.
if it is an autologous, that almost makes a little bit more sense.
But isn't Solaris' whole thing like they do autologous cell therapies?
Yes, but for mostly for CAR T therapies, I think so far.
Well, right.
I
But
don't.
what I mean is that like autologous is usually done at a CDMO,
Right. Yeah. Fair enough.
regardless of it being autologous.
Yeah, but for
Yeah.
something like this, right, where they're going after neurology, it might make a bit more sense. There's not the same urgency there is with like a cancer patient.
Sure. Yeah. Well, anyways, so RMAT designation. Is my camera frozen?
Just a little bit.
Oh my
Might
goodness.
have to shut off Claude for a second there.
It is shut off. My computer is just like
You're back now. You're good.
annoying. Okay. Well, anyways, that's that. I think we talked about Aspen like last week because they completed dosing. Yeah. I
That's
just
awesome.
I just think that I'm like a huge nerd for regenerative cell therapies in neurology.
Yeah.
Because who wouldn't be?
Excellent. Cool. All right, folks, we've got our about 8 minutes left before we're going to be speaking with Michael Torres. Stick around for that, for him. If you've got any questions for him, feel free to put them in the chat. We will get to them post-haste. But we're going to cover one more signal before he joins the show, and that is going to be a company called Selexion Therapeutics. Hopefully I am pronouncing that correctly. Selexion Therapeutics. The major signal here, if we just scroll down there for a second, Jeff, if you go to the is that they have dosed they've activated rather their first phase 2-3 trial, and in particular what I'd really like to focus on is just kind of the approach that they're taking here. So first of all, the disease that they're going after is advanced pancreatic cancer. So literally like the worst cancer at the very much end stages. So
Yeah, I'd say GBM, like glioblastoma and pancreatic cancer, are like the worst prognoses.
especially advanced, like it's basically like go home and hug your family and spend some time with them. Like you're out.
This is what Alex Trebek had.
Yeah, exactly. Alex Trebek, a very famous Canadian. But anyway, what
Justin
they are
Bieber, also a very famous Canadian.
we'll stick with the image of
Wayne
Alex
Gretzky.
Trebek. Shut up, Jeff. Okay, so anyway, what's really interesting here, Jeff, is that they are basically developing an siRNA therapy. Earlier on, folks, we were talking about saRNA therapies, right, where you're activating RNA. This one is a more standard siRNA where you're trying to inhibit. But what's really interesting here, Jeff, is that, again, they're going after like KRAS mutations. Can you tell me very quickly why KRAS is important inside of cancer?
Yeah, when it gets mutated, you just get it promotes proliferation. That's like the easiest explanation.
Exactly. Okay, so KRAS, super important in order for cancer to progress correctly. And so a lot of different therapeutics have been developed against KRAS, and obviously for very good reason, because it's such a big target inside of cancer progression. However, the problem with cancer is that it loves to mutate. And so a lot of these different therapeutics that go after KRAS can't get all of those different permutations of KRAS that are usually present inside of one patient versus another. And so it gets really, really complicated just because KRAS is so prevalent and there's so many different permutations of it. And so Selexion in particular have focused on these siRNAs that are a bit more generalized, that are basically able to mop up and catch all of these different permutations of KRAS and ideally do a much more effective job at stopping it.
Well, and the thing too here is that an siRNA targeting a mutated KRAS gene is not going to touch anything else. if it's specific enough, right? Like
Right.
usually an siRNA needs like basically 100% homology or complementarity to bind. So having it be against like, G12D, G12V, it's going to not touch like normal KRAS or anything else. What I think is interesting here is I wonder if they have like a cocktail that targets both G12D and G12V mutations, or if it's the same siRNA that can target both, and that single nucleotide is like, I don't know, what do they call it? Like, there's a name for
Base
it.
editing?
No, it's like loose or whatever. I don't know. But no, it's really cool. I don't know why. Again, like the cancer guy in me is like, why hasn't this been done before? Why are we just now starting to see that these approaches? Like we talked about that the saRNA from earlier, which I've never heard of, short activating RNAs, whatever. But to overexpress P21, like, yeah, why have we been doing that like 10 years ago? We've or using siRNA to knock down oncogenes. I never understood it. We're seeing them pop up more and more now, and it's got to be because of delivery or manufacturing or whatever, or like cell penetration technologies are advancing. But still, it's one of those things where like it's like it makes the most sense to do this kind of stuff.
Yeah. And the other thing that's really interesting about this approach as well, Jeff, is that they are going via a dual administration approach. So they're doing a direct injection, and they're also doing a systemic injection to just mop up any KRAS.
Yeah, you know why?
Why? Because it's
Metastasis.
Right?
Yeah.
So it's a really fascinating approach. So it's good to see that they're kind of going after intratumoral injection, but also systemic. So it's something that can be used for metastasizing parts of it, since it is advanced pancreatic cancer. Yeah. Anyway, excellent. So, folks, in just probably like 2 minutes here, Michael Torres has joined the green room, so we'll be bringing him on shortly. Looking forward to having a conversation with him about R10 and what he's been cooking up over there since his exit from Crossbridge. But yeah, just wanted to finish up here just with regards to Selexion. Yeah, it's currently in phase 2-3, so that's really exciting to see. And I think, again, Jeff, we were talking about this with Insilico medicines a little bit earlier, but again, this would probably be a really good candidate for combination therapies with other things as well. I'm blanking right now, but remember that really that other drug that made a huge splash.
Yeah, it was RevMeds.
Yeah, that was also in pancreatic. So one wonders if there could be a further benefit from combining those two together. Who knows?
You just love combination therapies.
I love
if this one works and that one works, just put them together.
Sink at biology, man. Like
exactly.
There's so
I enjoy rational combinations, not just like, well, the drug one work, drug two work. How about drug one plus drug two? Yeah.
Great. Okay. Fun. Well, we'll leave it there. Jeff, should we bring Michael into the studio?
Mm-hmm.
Excellent. All right. Very excited for this, ladies and gentlemen. If you have any questions for Michael, feel free to put them in the chat, and we'll try to get to them post haste. But without any further ado, let's bring him on the show. Michael, welcome to the Biotech Voyager.
Hello. Hey guys. This is fun. This is actually my first one, so I'm like,
oh, nice.
And I caught the several minutes of it, and yeah, I like how it's going. So you probably get a subscriber from me now. So
oh, nice. All right.
Oh, well, shucks. Thank you very
much. That makes one subscriber to our show. All one of you.
Congrats.
No.
Congrats.
Excellent.
Well, thanks for agreeing to come on. If it's your first one, we feel pretty privileged.
Yeah, no, I'm super excited, and I appreciate the opportunity to share kind of the Torres verse, a sliver of it, as I like to call it, having
nice,
completed my exit, by Eli Lilly and so forth and so on. So yeah, it's fun. So.
Right on. So I understand you've also got like an interview later today. I saw the LinkedIn post you put out. So you're on a double shift.
Well, and that one, I'm actually speaking on a panel at a ADC conference
Ah,
here in Boston. So
Oh,
that's
nice.
live, and I'm sort of covering all the modalities in which I'm interested
Yeah.
in developing drugs and TAs and things like that. So
Okay, because I was going to say this is not ADC conversation today,
this is
here
not.
at least. Yeah.
This is Arcan, longevity,
Yeah.
crypto, DeSci, all the fun stuff that
Nice.
I'm working on, and love to share more about that, of course.
right on. Perfect. Well, I guess just in case anyone isn't fully familiar with your background, can you give us a quick introduction, where you came from, what was happening over at Crossbridge? What's the story there? Like the condensed version.
Condensed. So I'll do the super fast condensed version. So, scientist by training, did my PhD at UT Southwestern in Dallas in cancer biology. I also had trained at the NCI. I had gotten a scholarship actually from the NIH during undergrad. And so backgrounds like DNA damage, DNA repair, genomic instability. And then in grad school, I got really into biotech stock trading and would use Twitter to talk about my ideas. So I have a pretty decent following and group, nice cohort of folks on Twitter or X, but I'm a Twitter lifer. And so I met a bunch of bankers, hedge fund guys, was writing reports for hedge funds in grad school. And then I wanted to do a biotech company myself in the cystic fibrosis space, which is an area that I'm personally invested in. And so that, fortunately, there was a professor there at UT Southwestern who had started a company in the past called Reata that was acquired last year by Biogen and was very entrepreneurial. So we started Recode Therapeutics, which is a genetic medicines company, raised over 450 million. I was on the board through 200 million raised. We had the venture arms of Pfizer, Sanofi, Amgen, and Bayer in that deal, along with big investors like OrbiMed and EcoR1, MPM, Vita Ventures, things like that. So that was a really nice entrance for me to like totally level up from the very beginning. Again, being on the board, but I wasn't part of the management team, and that was a little bit of some intrigue that I'll spare you guys later. I'll spare you guys from this. It's a fun story for sure. But long story short, I transitioned. Then I went to think about what to do next, and I joined this accelerator in Texas that's focused on cancer therapies. And from the local ecosystem, which we have, MD Anderson, UT Southwestern, Rice, Baylor. There's really good universities here. And part of my whole thesis of build with the buyer in mind is I diligenced some interesting projects, including this ADC linker technology that ended up becoming, Crossbridge. So I started that 3 years ago. It was academic papers. We did just one financing, essentially. It was a 10 million seed financing off the back of some, feedback from Pfizer Ventures on like what they'd like to see for the Series A. And then, instead of doing the 75 million Series A that I had lined up, we ended up being acquired by Eli Lilly in April. And then since then, right, like yes, I'm very fortunate. The deal was really great for everybody. And with that, I've been working on several other things, including Artan, which is a longevity company that's focused on, again, addressing aging, and can share more about that. But it's the unique thing is we funded it with crypto, so it has its own crypto token called Vita RNA that's got a several $1 million market cap. The token. And it's a really interesting way to fund science, early stage science. And, if you talk about it openly and it's very community based, you can create a win-win scenario that to be frank, is just more liquid than a private company like Recode, which I still own 1 million shares of, but it's private, doesn't do anything for me. But tokens and DeSci is a lot cooler, I will just tell you, and a lot more fun.
So I have from the whole thing. Congratulations! It's a very impressive resume. The one thing that caught my attention at first was you were able to be investing in biotech stocks on a grad student's salary.
Yes.
Or was it just like an interest? Because I'll tell you what, I did not have the means in grad school to be investing in anything besides like rice and hot sauce at the grocery store. So that's interesting just off the bat.
I had
also, how do you go from there to writing reports for hedge funds while in grad school? What's the
You must have been pretty good at
it.
well, so a couple of things. One, I had a few good wins actually that kind of set the foundation for me to then continue investing. And then each report that I wrote was like $3,000 to $5,000 per report, which was over 10% of my grad school.
I was going to say it's like 5
so,
times your salary.
Yes, that sort of really opened up a lot of doors. And I'm not a trust fund kid or anything, so it's not like I had all of that money. I mean, I hear you on the ramen and everything like that. Living the life during graduate school.
Eating peanuts, as they say, for dinner.
So yeah, I mean, I think that was again just really opened the door for me to get really interested in doing biotech that way. So.
Fantastic. Right on.
Well, I have a lot of questions on Artan, but I can wait until we get into that conversation.
I think we can. Yeah, let's jump into it. Well, I guess let's back up for a second here. So so with regards to the idea for R10, like what was the initial thought here? What was the problem that you saw that you'd like R10 to be able to solve?
Yeah,
like I know that there's like longevity in general, but like what was the particular mechanism that was of interest?
Yeah, so it's actually an interesting story as we were working on Recode and Recode again was my postdoc project that I started. So I've been doing academic work after I graduated, and it sort of morphed into Recode. And the first approach that we went after within Recode was trying to address nonsense mutations, which are these stop mutations. You don't make the protein, and in the case of cystic fibrosis, there was a large fraction of patients that had these nonsense mutations, and the drugs that Vertex at the time were just getting approved for, which has made Vertex the behemoth that it is, they didn't work on these proteins because they're all like help fold and correct CFTR, the protein that's defective CF, and so I got really interested in trying to correct nonsense mutations. And so with that, there was this really interesting biology around a particular codon for an amino acid called arginine. And so arginine has, I think, 6 codons that can code for it, but there's one that's very rare, and it's actually underrepresented compared to other codons out there. And the implication of that is it's something that we've been trying to evolve out, but arginine is a functional amino acid, so we arginines are very prevalent. And so in this case, that rare codon is a CGA codon. And so what's unique about this CGA codon is that a single base change from a C, which is cytosine, to a T, then makes a stop codon, and which is bad. You don't want stop codons. And so if you look across all disease-causing nonsense mutations, arginine is actually the most common. And then on top of that, if you look where arginine CGA codons are found, there's a whole host of proteins that are involved in age-related biology, if you will. So DNA repair proteins, tumor suppressors, proteins involved in like neuroprotection, so like APP, things involved in like Parkinson's, Alzheimer's, like I just said, and then actually proteins involved in a blood aging process or disease, if you will, called CHIP. So that's you get this like clonal expansion of less than ideal functional blood cells. And so collectively, the whole premise was, if we could restore the proteins that go down over time. And the other point I forgot to mention is that, again, cytosine is a very commonly mutated nucleotide. So, it can be deaminated, it can be methylated, and we hear a lot about like methylation clocks in aging, and that also sort of reduces expression levels over time. And so, but methylation also sensitizes to deamination, and that's a chemical change that then takes that C to a U, as I mentioned, and so but there's also like UV exposure causes C to T transitions. There's a whole host. So again,
I think C to T is the most common mutation.
Yes, exactly.
Period.
Yes. And so, in spite of all the cool guys out there doing, partial reprogramming and blah, 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 and all kinds of other stuff,
you get the label of
biology, that we're going after that nobody, is, and we think it can have huge implications across multiple disease areas. And maybe the last thing I'll say, and then, I'd love to hear you guys' feedback, because I wanted to just layer in one more part about the biology. If you look for diseases that have, say for instance, like ATM mutations, ERCC6, the XPD, these are all proteins involved in DNA repair. There are rare diseases where people will have nonsense mutations. They're homozygous for these in most cases. Besides, cancer risk, one of the hallmarks of the disease is accelerated aging. So again, a lot of the progeria also have this phenotype. But speaking to, like, if you could suppress that, and we have this technology that we're using to restore the protein that's lost there, the amino acid arginine, it could have a really profound effect on aging. And so somatic mutations is the other sort of word and buzz phrase that people use. And that's what we're trying to go after.
So just so you know, Michael, you get the label of cool guy, number one, because you've had an awesome history, and this science is cool. tRNA therapeutics is really, really interesting. So like it's, cool is definitely a thing that I would call it. So the one question that I have is you're basically correcting these stop codon mutations. UAG in particular.
UGA,
right?
Yes, UGA.
UGA, sorry, UGA.
UAG is a stop codon. So
yes, there's UA. I learned them like this. There's you are arrested, you are grounded, and you got arrested. UAA, UAG, UGA. So UGA, right? But if you are giving a UGA tRNA with an arginine, wouldn't you be potentially giving normal stop codons? Wouldn't you be replacing those with arginines?
Yeah. So
how do you distinguish between a pathological stop codon and a normal UGA stop codon?
Yeah. So the beauty of biology is biology has done that for us, right? And so so the details around that is, typically for normal, like ending of translation, there's usually multiple stop codons in frame, and they're usually different stop codons. Okay, so the high level version is a normal stop and a premature stop look completely different in terms of like the complexes that are around for to finish translation. There's this whole like sort of loop type thing that happens with the 3' UTRs, and so like the ribosome is designed to like fall off as it gets to the end of a normal message, and so just having that tRNA present doesn't like stabilize that and make it read through. And we've actually, and others have looked at this too, using ribosomal profiling, which is a very like sensitive like assay to look if you're reading through normal stops. And so in our case, again, we've looked globally, and this was done with Rachel Green at Johns Hopkins, where we didn't see this read through. Now, if you take a small molecule drug that can help promote read through, like aminoglycosides are like the most famous version of these drugs. What they do is they loosen kind of like they bind to the ribosome and loosen translation like fidelity. And so in that case, the ribosome does can stay on, and you can see with some of these drugs, a lot these ribosomes in the 3' UTR, meaning that they've read through the stop. So I think it's
okay.
So that's one of the sort of supporting evidence that a tRNA doesn't read through normal stops. And we've done some early animal studies, and then we're gearing up to do a monkey tox study later, and then ultimately, right, we want to be in the clinic and see how this.
So basically, if we understand correctly, for a normal stop codon that is completely healthy and should be there, there's basically just so much context that it's coming. And so, like, I don't know, you're watching a movie or reading a book, that the end of the book is at the end, and there's a bunch of context and the stories kind of leading up to that. And there's a bunch of additional information that you can pull to understand this is real, versus just randomly a stop codon in the middle of the book. Something that's just kind of reading the text might not figure that out. But for something like for tRNA biology, we have this ability to read that context and figure out that isn't at the end of a book. That isn't at the end of a movie.
Yep. Exactly. So again, there's nature has designed big giant stop signs and a lot of the scenery changes when you're getting towards the end, and then there's these termination factors. There's all these proteins that are there ready to, like, kind of stop. Where in the again, in the middle of a message that's not present.
So then how does the aberrant nonsense mutation actually terminate translation properly?
How does it aberrant? Because in that case, right, it does trigger other factors that then will the most common way is a stop codon by itself has like a braking ability, like
Sure.
like brake, pump the brakes, and then it'll trigger and forgive me for not remembering all of the biology, but it's subject that you normally get nonsense mediated mRNA decay. So in that case, right, there's these proteins, I think like UPF1. I'm trying to remember all of them that then sort of surveil for these things, and then you'll get the message to say, hey, there's a stop, this is wrong, and then it the message. And so in the case of with the tRNA, it actually stabilizes translation, and then that stabilizes RNA, the mRNA as well, and then you can continue to like proceed.
So it's more biologically complex than just like erasing a period.
Yeah,
your technology, because if you're going to go around erasing periods, you're going to get a bunch of run-on sentences. That's kind of what I was thinking about, right? You might allow the improperly truncated sentences to continue to be translated, but you'll also get a bunch of run-on sentences, and you're going to get a bunch of like protein degradation and like just misfolded proteins that you would need. But if there's more context involved, and like the end of the translation process doesn't wholly depend on the presence of the stop codon there, then it wouldn't be that big of a deal.
And we'll see what happens. But that's
sure. But I've seen other companies doing the same kinds of things where they have like a tRNA to correct for nonsense mutations. Yep. And every time I see it, I'm like, how does this not cause death? Like what? But there you go. That was my one question that I had. I was like, how are you not going to cause death, Michael?
Well, I can't tell you that yet, that we haven't. So let's cross our fingers. Whatever else, but I think we're pioneers, but not completely the first in this. Because to your point, there are other companies that are have been thinking about tRNA based therapies, and so
awesome. Fantastic. Cool. Okay, so a little bit just about like the mechanism. So how is this whole therapy getting delivered? Is it fair to call it like a standard gene therapy?
So in our case, we're thinking about multiple different delivery vehicles to address like various tissues that we think are going to be the most important for this therapy. And so right now, because we do think we need long-term expression, we have been thinking about a viral vector, and in our case, we want to hit as many tissues as we can. So we are looking at using AAV9, which actually has very broad tissue tropism. And we have some mouse data that shows that as well. And, of course, one of the everybody talks about safety, and we've certainly seen even recently, right, some safety challenges with AAV9. I think that was probably more on the CMC side and not sort of doing what you needed to there. But, in our case, tRNA is a natural occurring molecule in cells. It also is a very highly abundant molecule. And with the exception of, I'd say, like immunogenicity with the capsid itself, we use a different promoter system than what mRNA. It's POL3 versus POL2. So there's all of these, like, I think nuances that we hope will ultimately translate into needing lower doses of our tRNA to be therapeutically relevant. That would obviously then help with the safety profile of our drug. And so that is like one delivery vehicle that we're working on. We're actually partnered with a really cool company called Cyanex that's based in Chicago. Josh Leonard's lab.
Shout out Jay.
Yeah, Jay's great. And he's in Boston now too. So he had a board meeting yesterday, and I invested in this company. So full disclosure.
Nice.
I was
but they're awesome.
Yeah, I was a customer first. So I think, we should put that on the website. But so we're using his engineered EV platform to go after HSCs, because we think HSC, again, I mentioned this disorder called CHIP, clonal hematopoiesis of indeterminate potential, that is actually age-related. AAV9 doesn't hit HSCs, naturally. So we've teamed up with Jay to work on HSC targeting and are moving that forward. And because hopefully in the next few minutes, we'll talk about like the crypto angle of all of this. We're actually now working on a cosmetic version of this, of our tRNA that we could package in a way that, we could create a product as early, create a product that then again with crypto and things like that, people like to see, products and real world assets. We know drug development takes a long time, but it's still using that same biology in our skin cells. As I mentioned, UV is one of the main triggers of this. I think it's like the SSB1. There's all of these names for these types of mutations, but the C to T transition. So we have like a microneedle device thing that we're working on right now. And hopefully towards the end of this year, early next year, we'll have that all finished out. So there's basically 3 types of programs that we're doing within our TAN right now.
What is the indication in the cosmetic area? Like wrinkles?
No, I mean, indication in cosmetics, you can't really have an indication in cosmetics because then it's
ugliness is the indication.
It's regulated, it's age defying. It's so there is a balance between hardcore like biology and proof points, and that then says this is a regulated drug and you need to go do that. And I mean, I live in a regulated world, and I'm not opposed to that, of course. Which is why, the claims are very important. Like what you say something can and can't do in the cosmetic space is very, very, carefully worded. And this is why you can have exosome products and peptides and all kinds of stuff like that people are trying. And you can't have too much evidence, or else they can't market it at Walgreens or whatever.
That's interesting. Got it.
Yes. There's a whole world within that whole regulatory world there.
Okay. Would it be because we know what it doesn't do as well now? We've done so much testing that we know we can't say these things. Is that what it is?
Again, it's like you can't claim certain. You can't claim it's actually doing something without that needing to go to FDA to get approval to market that claim.
that claim.
But if you say, it's age defying or maybe wrinkle reducing might be okay. Basically, but at the same time, half this stuff really is not doing too much anyway. But we have real biology that we're addressing. So it is like, we're still navigating that full transparency there, but we think there is a way to kind of have a marketed product that can actually do something. It just can't do too much of something, because then it'll be longer to get in a different approval process. But I think as the biology plays out, I mean, we're totally willing to like go that route, because in a conversation, can't make any promises. We think that this could potentially be really effective in reducing like melanoma risk, as an example, if those like damage, the things that we're doing, the damage that UV is doing, if we could suppress that with our drug, like it potentially could keep, melanoma risk down, cells happy, collagen, elastin being expressed, all of these things that you want. And so, we're certainly not opposed to, thinking that this could really be a useful tool for combating, certain cancers as well.
Excellent. Okay, I know we've only got you about for about 5 minutes here, so I want to be really respectful of your time. So yes,
crypto.
Really excited by this whole concept of decentralized investment into science. So yeah, just kind of walk us through how that works. And folks, if you have any questions, put them in the chat and we'll try to get to one at the end.
Yeah, and so it's an interesting story for me, and I'll tell you how I got involved in it, and then what we've been doing. So post my Recode, Recode, I was connecting with some of my friends at Pfizer Ventures, and Pfizer actually had put in $500,000 into this crypto longevity DAO called Vita DAO, and Vita DAO's an organization that I think they raised about $4 million to then invest in various concepts in like for longevity. And so that's how I got even aware of like any of this stuff was through Pfizer, who put in some money, Pfizer Ventures, who put in some money into this into Vita DAO, and so with that, I got interested, because they're like, look, they know, like, they're looking at cool science, but they don't have any drug developers, people who know investors, people who have built companies like you. Maybe you can get involved. And Vita DAO has a token called Vita, VITA, and that's like on Ethereum, and I think that's on Base. And anyways, there's sort of like a governance token where token holders can vote on projects that they want to fund, and sort of
that's cool.
Have a voice with the token. And so in that case, they were looking for projects to invest in, and so I had submitted Artan to get funding, and originally we raised about $100K. So again, I didn't have any data for Artan at the time, and I said, look, I've got an idea. Here's the experimental plan. We're going to design these vectors. We're going to make them. We're going to transfect them in cells that have the nonsense mutations, and we're going to look for activity, right? And we did all of that with $100K, and then with that, then we raised $300,000 on as a token offering, and that was for the token Vita RNA. So that's a token that's out there now. That's again on Ethereum that people can buy. And in our case, what we did is we tied the token to essentially a sponsored research agreement. So with a middle entity, and I mean, we can go into like the technical details, but again, there was like a middle entity that then gave Artan $300K, and then we did a deal with the middle entity that then would give them rights to the drug, like you would like a tech transfer office. You get some royalty, milestone, things like that, and then they would that middle entity connects with the token holders to then distribute that as needed. And so in that case, there is economics tied to the token, and and we share the data openly, things obviously not for, like patents and stuff like that, but, we're very open with the data. And in that case, token holders can sort of place bets, if you will, on the trajectory that we're going to be going. And as we move through the drug development, pathway, the token has gone up, and we speak, and doing this. I don't know the token,
That's
but
cool.
maybe people will hear about it through you. But I think it's a really cool way to fund science. In my case, right, I have a token allocation as well. It vests, so it's not like we're rug pulling anybody or anything. So again, I wanted the sort of bottom line is we're doing everything we can to create this ecosystem where folks can fund science directly, but also get upside in like the drug development parts of that, and then ultimately, right, continue to support this ecosystem. And I'd say what also is really interesting is in our case, right, all of this data that I've generated, we haven't needed to do any dilutive financing at the same time. So I'm not diluting Artan Bio by like the cap table on Artan.
Right.
Even we're raising money on the token, and we can continue to raise on the token, and the token holders are happy and grow. Cool. And then
that's cool. So I was gonna compare it to like kind of like crowdfunding, sort of, but instead of like go investing in a company, you're kind of like buying a tokens. But that's completely different then, because when you go to like WeFunder is on your cap table, right?
Yeah.
If this isn't, how does that work then? Like you giving them royalties, you said?
Well, I guess just like, Michael, do you have a couple extra minutes? We want to be respectful.
Yeah. I've got extra minutes. Yeah. So I mean, it is an equity. I mean, again, this is where the token and the governance sort of comes into play. So and so we do have an agreement. Like, again, it's like a sponsored research agreement that ties to some economics. But reality is, and maybe I'll expand on this a bit further for the crypto parts. Like, crypto guys, they're incentivized, guys is general, but like they're incentivized by a lot of different things, and it remains to be seen, if they have the sort of think about the same profile as like like an equity that you hope in 10 years you get something out of it, unless you invest in me and Crossbridge and the stuff that I do. Then it's 3 years to exit right. For a lot of money. So everybody should just invest in what I do, man. I'll throw that out there. But so I think we're within Artan and the crypto communities and this platform. There's a platform called Molecule and another group called Bioprotocol that was backed by KDT and North Pond and other investors. They're the ones sort of creating this a platform in which science can be funded and go forward. Is continuing thinking of ways to incentivize token holders to like either hold the token or invest in fund. And so one way that we're trying to continue to expand that is what's called like a coin to company model. And so in this case, token holders could stake their token to then potentially get equity through an SPV, and so that now they could be on the cap table through the SPV. And if they want to continue to ride like the upside through what the company is doing, then they also have that ability as well. And then if you're doing like token burns or things like that, then you're reducing the supply. So then the token price goes up as well. And token holders who haven't done that are happy. Everybody's happy. So there's some really cool like financial things that are going on. And in the future, again, post Crossbridge exit, right? I talked to a lot of investors, a lot of funds, and I'm telling them, hey, I'm doing science and crypto. We got tokens. So investors are actually very interested in this because imagine a world where they could invest in a company, they get their equity, but then there's also a token out there, and they get an allocation of the token. And so if that token is liquid and people are buying and selling it, that's a way for liquidity for investors that doesn't exist today. they still like can ride out their equity investment. So we're working on building that framework as well. So I think, it's sort of like a whole brave new world there.
This is super cool. I'm totally not into crypto, but this might actually make me interested in crypto.
Yeah, well,
this is really interesting. I'm looking at this molecule.xyz website. It's super cool. It's clicking now. And there's this huge problem with the valley of death with early stage biotech. It's a new and interesting way of it's still kind of crowdfunding-y in my head. But it's not. It's different. This is super cool. Okay, I'll have to look into this more.
And no, definitely really cool. And not. I mean, you guys, you can just look up like my token or what have you. But not to talk economics there. But I mean, I think it was up like 1,200% in the last couple months. So it's
where do we? How do we find that?
Yeah, so the token is called Vita RNA. But I mean, we have a website, I think vitarna.xyz, and it's on the molecule page, and they sort of walk you through if you want to buy any of the tokens. I'm not over here promoting my token or anything, but all of that there, that ecosystem and that exists. So, you need a wallet. You have to kind of know crypto a bit, and, do you know how to swap between Ethereum to, RNA and so forth and so on. So I'm also trying to help the crypto guys. We need to make this easier for people to buy tokens if they want to support these companies. So yeah, I'm actually, I think as a concept, think this is a really cool new way of funding early stage science that better aligns incentives. So yeah, happy to take a few of these questions if you guys want me to.
Okay, yes, there is Sandeep's question I see is a little bit older, but I think we kind of did get through that a little bit earlier in the interview, Sandeep, so feel free to go back just in interest of time. Just curious, how does one prove functional efficacy on aging itself for a product like this addressing in a clinical study? Is it mostly just biomarker readouts? Good question. Yeah, what are the endpoints for aging?
Yeah, I mean, I think it's obviously a very important question that the entire field is trying to solve, right? Survival studies doesn't make sense for anybody, and we're too impatient to be okay, let's wait 70 plus years to see if this drug adds 5 or 10, right? So it's going to have to be biomarker driven, right? I think in our case, I'll give you examples how we're thinking about it. In this case of the disease CHIP, it's actually a very easy readout because you see this you can detect these cells that have this variable allele frequency is what it's called. And so if we are slowing that down or addressing that, we should see the populations of cells shift to show that we are doing something impacting in biology. We could potentially do a proteomic approach, right? Because again, over time, these proteins should be going down, and we know where the arginine, CGA, where those are found. So you can build a panel, right, to look either at the RNA level or at the protein level to see that we're sort of bending the curve as it comes to the amount of CGA or UGAs that you find, but really it's probably the protein levels that you would expect to go down. Yeah. And then, again, as I mentioned at the beginning, a common strategy, right, is for folks to think about some rare disease that, you would go after that then you can build the case for. I'm, not opposed to that, but I'm a big, blue sky, let's go for gold type thing. But, there is a world where, again, as I mentioned, some of these rare diseases that have a nonsense mutation in, a very a DNA repair protein, as an example, right? We might be able to see those age-related phenotypes be addressed in that rare disease. And, again, I think you have to sort of build the narrative that, okay, you are addressing this biology, and that biology then is involved in aging. And so then if you are, there's a good chance that this is could be a longevity drug. And maybe just to say quickly, there are, geographies and regulatory environments like, the UAE, United Arab Emirates, is going all in on longevity and trying to make it easier for these drugs to be approved and actually, get conditional marketing, very, very rapid as well. So I think, my whole premise is if we could show that we're addressing this biology in some way within humans, I think we can build the case that this could be a longevity drug, or or this could form the basis of multiple drugs, and then we'll let the market decide what they think the value is there.
Yeah. And I think of therapeutics involved in longevity isn't like aging isn't a pathology. There's diseases associated with aging that you would be going after, right?
Yeah.
So it isn't like, oh, we're just going to stop aging. Like that's not what you're doing.
Yeah, but I mean, I would say I think it is, something that is addressable now to extent and how long, and then you can get all futuristic about it. But I think
stopping time itself.
Yeah, I think we all want to live healthier and longer. And I think, the GLP-1 class is the perfect example, and people point to it now of like something that is likely to significantly improve health span and lifespan. It's not going to like improve the the ceiling perhaps, but we're going to live healthier longer, and that I think is now seeping into the conversation where there's other drugs that we're saying could be useful to do similar things. And that's obviously the whole point behind like partial reprogramming. Hey, we're going to make your immune cells last longer, better, so you're going to be healthier over time, and so forth and so on. So I think the conversation is starting to shift, and obviously I want to be on the forefront of that as well, and do it in as many cool ways as possible, and so we'll see what happens. So
phenomenal.
Looking forward to seeing it. Yeah.
Awesome.
Cool.
Well, Michael, you gave us an extra 12 minutes. Really appreciate your time. Very excited to see where you guys go from here. And just so glad that you've made the switch from bispecifics to gene therapy.
Dude, I have 4 companies I'm working on. 2 have already been funded. So and some are in biologics. Again, we talked about some delivery stuff as well. So I'm in the mix, man. So so yeah, always happy to chat with folks. I'm pretty accessible. You guys know that as well. So cool. All right. Well, thanks for having me.
Thank you so much.
Thanks
really appreciate your time.
Yeah.
Thanks for coming. Right on. Well, Jeff, that was super cool. I'm glad that you're potentially interested in crypto now.
Yeah, that's like, it's one of those things where when I think of cryptocurrency, like the vast majority of them, I'm not gonna lie to you, I immediately just go, it's a scam. Like it's some kind of like pump and dump scam, whatever. Right. But this seems really, the word cool is like the only one that's coming to mind. Yeah. But it's just because it's like, oh, this is like an interesting new, like young way of funding science.
Yeah.
And like even like the like this Molecule XYZ website, like this is a cool like idea.
Can you share quick and we can look at it? You're just
still
sharing the Biotech Voyager.
Yeah, let me pull it up here. And then if you go to, let's go to theirs, Vita RNA. And from what I'm gathering, okay, like this might not be completely accurate, but from what I'm gathering, it's like you buy this token, okay? You buy this cryptocurrency token, and this token, this Vita token, is dedicated to using that money to fund projects.
Exactly.
Right? And it's not just like, because most cryptocurrency is just like this ethereal, like it's worth a lot because people think it's worth a lot. Right? It doesn't actually do anything. But this isn't that.
Yeah.
But I'm still wondering, like, at some point, money is going to Artan Bio. And then somehow, some way, that money is increasing and going back to token holders.
Yes, like the value of it goes up over time as there's increased demand. It's supply and demand, right? You release a certain amount of the tokens, they're available on the market for sale. as more people buy it, and as more people as the research that the company is doing creates more interest and gets more press or good results come through, that increases demand for the token as well. So the value of the tokens that you've bought go up.
But let's say that I go and I put in $100,000 into this token,
right?
And then it goes up, let's say just like 2%,
right?
I want to cash out. That $100,000 doesn't exist. Like Artan Bio's bought antibodies and like done a mouse study with it.
So I think that's where like the
third party will eat it.
The third party comes in. Yeah.
Yeah. The third party, the middleman will kind of eat that and say, we have $50 million and we can kind of have this as expungible stuff.
Exactly.
Okay. Yeah. I mean, it's super cool. It does read a little bit like crowdfunding, but there's this like cryptocurrency aspect that might make it more accessible or like different financially than just like, I'm going to invest in WeFunder.
Yeah. Well, and again,
which is the cool part.
It allows anybody from across the world. So there's not the same like regulatory limits, right? So for example, I was super excited by Siren Biotechnology when they did their crowdfunding last year. I wanted to invest. Sadly, I'm Canadian, so I couldn't. It was only for US residents. This is something that allows you to draw in liquidity from across the world from anybody who's got a phone and sees the value.
Right.
So I think it is an untapped way to kind of go after investment instead of focusing on, trying to get one person to sign you a check for $5 million. You can crowdsource from the entire world's population of people with a phone and a couple bucks to spend.
Yeah.
To kind of get an early stake into your company that grows with time.
I wonder if this company Molecule would want to partner with the Biotech Voyager it seems like a very similar, like early stage biotech companies looking to crowdfund, quote unquote crowdfund. Like, perhaps. This is cool. It's a super immature.
Should we get a crypto sponsor?
We can just make our own like Voyager coin. Oh gosh, it gets like so complicated in terms of like legal. Yeah, we'll call it Vogue coin.
Vogue coin.
All right. Anyways,
excellent.
What do you say?
That was a great show, folks. Well, first of all, if you just go back to the Biotech Voyager, folks, if you're not subscribed to the Biotech Voyager YouTube page where you are right now, we would greatly appreciate it if you liked and subscribed. Also, check out the biotechvoyager.com, completely free, and we have a ranked list of the hottest early stage biotech companies. It is updated, it is live, and also you can filter it by the different companies that you're interested in. So if you want to look at cell therapies and AI, you can literally do that, and it'll do a ranked list again of like the hottest companies inside of the space. Yeah. And search. 3T Biosciences, Anaka, right up there. So Create Medicines is in there somehow. That's an imposter.
Well, it is a cell therapy.
It is, I suppose.
Kind of. If CAR T cell is a gene therapy, then I think that in vivo CAR T cell can be a cell therapy. It's intermixing.
Right on. I guess the last thing, if you want to leave a comment or in the chat, who should we have on next? We both have fairly large networks, and if there's someone in particular that you would like to see us bring on the show, do let us know, and we can try to get to them. We are planning on featuring more and more early stage companies, and so if you guys have any suggestions on who we should bring on for an interview, do let us know. We've got a nice roster of people coming, but always looking for more suggestions. Excellent. All right, Jeff, we'll leave it there. Thanks for coming, folks. And we'll see you later. Bye now.